which, I've kind of been thinking about for a long time. One of the things, and we'll go into this a little bit, is that, autism has been thought to be highly genetic because it's highly heritable. So heritability has been equated with genetics, absolutely. And that's because, genetic diseases are inherited, and that's what we think about for genetics. But of course, we know the numbers don't add up in the genetics. So I want to introduce the idea that there may be inheritable disorders that may not be fully genetic. They may have genetic environmental influences or due to the environment that people are living in. And it's kind of a shift, and it opens us our idea of where we can look for etiologies. So again, my disclaimer, any treatments that I talk about for the most part are not FDA approved. They're used off-label, and only practitioners who have the knowledge of the risks and benefit of these treatments should use them. So the heritability of autism we have, has come from this idea that identical twins have a much higher concordance rate than fraternal twins, and fraternal twins have a higher rate than non-related individuals. And so this definitely makes an argument for heritability, but this is an automatic conclusion that this is a contribution of genetics. And I don't know that that's something you can make automatically. So the heritability of autism is another study that shows that it's about 81%. And so that's a lot. And so again, in this sentence, again, they talk about inherited genetic factors. And the fact is that, that there's no evidence for that. They're just assuming that it's heritable, that it's genetic. And then when we look at genetics, and this is kind of where things don't add up, to look at the genetic inherited or the genetic abnormalities, in fact, we find that Mendelian, so inherited, single gene mutations only make about 3%. Chromosomes abnormalities and copy number variations, think about 5 to 10%. And the large part of them are de novo single gene mutations. So what does de novo mean? De novo means that it's not inherited. It wasn't in the parents, okay? So, I like to describe, I hear all these genetic talks, and they talk about, well, this is genetic because there's this high heritability rate. And at the end, they talk about whole exome sequencing, and they say, well, we know this is the gene because it's de novo. So it sounds like somebody's drinking the Kool-Aid a little bit. So I just, and it's not to put down genetics, it's just that there's other factors that we, have to think about, So then you have healthy man, healthy mom, healthy dad, and they have this child with uninherited mutations for the most part. So the idea that the heritability is due to genetics just doesn't add up. The numbers don't add up. This is a very known study by Hallmeyer that shows in a twin study, genetic risk factors, when you look at genetic risk factors for autism, is 38%, whereas the other risk factors, environmental epigenetics, environmental gene interactions, was what made up the bulk of the variance there, suggesting that, genetics does play a role, and we know many syndromes that are related to autism. Of course, one of the problems is we haven't found the autism gene because there's hundreds of genes that seem to be related to autism instead of just one. So we have to kind of look at this in a larger sense that you have genetic susceptibility, but you have all these different modifiers, right? You have epigenetics, which we're not very good right now at detecting these changes. We have environmental factors. We have sex-linked modifiers and hormones in utero that we know can modify development. So those are just some of the things that may be non-genetic. And so for this, and the way that I kind of thought about this, and recently is some of the research that we've been doing, where we're finding that some of these metabolic disorders that we're looking at seem to be inherited for some reason, but there's no genetic basis for them. So I'll talk about the first disorder that I've been working on for a while, called the cerebral folate deficiency, the folate receptor alpha, and what we found out about transgenerational transmission. So cerebral folate deficiency, to quickly go over it, is was described really formally in 2005 by Dr. Rainmakers and Dr. Quatros, who were really the leaders here. And what they found is that certain kids with neurodevelopmental disorders were not getting folate into the brain. Dr. Rainmakers thought this was genetic at first, sequenced the folate receptor alpha, the main transporter mechanism that brings folate into the brain, and he couldn't find any mutations. Dr. Quatros took a look, and he found these autoantibodies that hang on the folate receptor and make things, make it not work very well. In this first paper, they described kids with neurodevelopmental regression, decreasing head growth, so acquired microcephaly, dyskinesias, movement disorders, and epilepsy starting at a young age, usually about 1 year of age. And so here is the data where, this is the first paper they introduced, these antibodies. Dr. Quatros found these antibodies, and we can see that the patients with cerebral folate deficiency have very high titers, and control subjects have near zero titers. And how does this work? Well, everything that gets in the brain has to get there through some type of transporter for the most part. And the folate is no exception. So folate hangs on to the folate receptor alpha, and it's transported across the choroid plexus and dumped into the CSF. Well, Dr. Quatros found two antibodies, the blocking antibody here, which blocks the ability of folate to bind to this receptor, and the binding antibody, which binds to the receptor and just messes with how it works. Luckily, we have a reserve, a backup system called the reduced folate carrier, which we can use therapeutically to get folate into the brain if we identify these children. So as more and more case studies were being published about children with cerebral folate deficiency, there was many papers publishing that these kids had autistic-like symptoms. So myself and Dr. Rosignol saw this as an opportunity and said, well, we can measure this biomarker. How many kids in our clinic are walking around, how many kids with autism in our clinic are walking around with these antibodies that we might be able to treat? And we thought, well, it was thought at that time that cerebral folate deficiency was a rare disorder, so we thought, well, maybe it's a couple. And so what we found is that in the kids in our clinic, that 60% of them had the blocking antibody and 50% of them had the binding antibody. We went through, we did an open label trial, and then we did, this is a double-blind placebo-controlled trial we did at Arkansas, along with John Slattery and Shannon Rose, who are both here, who were instrumental in this trial. And we showed that using leucovorin or high-dose folinic acid improved language significantly in 12 weeks in kids with autism. And the thing that predicted that was the folate receptor antibody. So then Dr. Quatros, started looking at families, and he found this data that was really interesting. When he looked at siblings of kids with autism, that he found that there is also a high rate of the antibody, and he looked at parents, he found out the parents had these antibodies also. But when he looked at controls, it was present in a very low level. So this suggested that somehow there was something about this where they seem to run in families where the whole family may have these antibodies. And why is that important? Because the same mechanisms that bring folate into the brain across the blood-brain barrier also bring folate across the placenta. So if mom has these antibodies, the kiddo might have a double hit. They may have problems getting folate, before birth while they're developing, and then folate problems after birth. And it seems like this mechanism may also affect the father, and that's because probably because the folate receptor alpha is also on the epididymis, so it may affect actually sperm development. So Dr. Quatros actually developed a model, a rat model, where he injected the folate receptor antibodies into the rats. And the pups he looked at, and these are different tests, passive avoidance, active avoidance, and such, and he showed that those with saline had perfect performance, whereas those that were given exposed either during gestation or postnatally, weaning and getting it through the milk, had much poorer performance than the other rats, the control rats, and they had characteristics that looked like autism. This is another study where he showed that actually on the placenta in the rats, there was actually inflammation by the folate receptor alpha, and that there was problems with flux of folate through the placenta. And then in this paper in molecular psychiatry, he treated them with folinic acid and showed that you can restore their behavior and rescue these babies if they're exposed to this antibody in utero. And the important thing here is that the treatment is folinic acid, leucovorin, a type of what we call reduced folate, which is more bioavailable than folic acid. And folic acid is the oxidized form of folate that a lot of people have problems metabolizing, actually won't go through the transporter. So the idea that we give high doses of folic acid to women to try and possibly fix something like this may be a little bit misguided, and we may be wanting to give actually some of the more reduced folates, which are more bioavailable and go through the folate pathway better. This is an interesting study we did where we found that the folate receptor antibodies seem to correlate with a thyroid stimulating hormone. And we thought, well, why is that? Because there's no folate receptor alpha on the thyroid, so how could this antibody be messing with the thyroid? And what we did is we actually got fetal tissue, and we found out that there are folate receptor alpha receptors on the thyroid and prenatally at 15 and 17 weeks of age. And so what we hypothesized is mom has these antibodies during gestation. This will actually interfere with development of the thyroid early on in gestation. Dr. Rainmakers did the study where he looked at families where either the child was positive or the mother and father were positive. And what he showed, you can see here, this is kind of goes up as you have more positive people, that the severity, and so this is the CARS score, which is a measure of autism symptoms. So we'll see, look at the blue, and we see for the most part, the more individuals in the family that have the antibody, the more severely affected the child is. And this is the orange bar is after treatment with leucovorin. There's actually a better response in those that have fewer antibodies. If you have more antibodies here in the family, this response becomes non-significant. And so there's this kind of model where, you know, if you have mother and father positive, you may have very severe autism with a poor prognosis. Mother only positive, you may have severe autism, and father, you have moderate autism with a better prognosis. Of course, this is kind of just from very preliminary data that we see, so we have to replicate this. And I won't go over that. So that's kind of interesting that we're actually finding that these antibodies, which seem to be important and help us in treatment, are actually somehow inherited in the family. There seems to be families that have them and families that don't. What's the mechanism? You know, we still don't know what it is. And we're looking into it, but it seems to be, at least for now, some type of non-genetic inherited factor. So this is another, this is recent data we found in mitochondrial dysfunction. So, I don't know what's happening here. So the mitochondria is the powerhouse of the cell. We have anywhere from hundreds to tens of thousands of mitochondria per cell, and they're very important. Really, mitochondrial disorders were first described in 1988. When you have mitochondrial problems, they affect high energy dependent tissues like the neurologic system, gastrointestinal system, and immune system. And these are what we see in many kids with autism. So it's no surprise that we find mitochondrial disorders in autism. When we started to look at this, the first thing we did was say, well, how many kids have mitochondrial disorders in autism? And so myself and Dr. Rosbuell did this meta-analysis and systematic review, and we found that the studies bared out that mitochondrial disease, which is a disease where mitochondria is functioning very, very poorly, is seen in about 5% of kids, but then up to 30% of kids had things like elevated lactate, which demonstrated the mitochondria wasn't working very well. So there was kind of discrepancy between this 5% and this 30%. And so we decided to look at this in more detail, and we also wanted to look at the interaction between oxidative stress, which we see in kiddos with autism, and the mitochondria because they're very interrelated. We used something called the seahorse, which is a high throughput respirometer, and a lot of this was really championed by Dr. Rose at Arkansas Children's in the lab to develop this assay. And we measure these different measures of mitochondrial function, basal respiration, ATP-linked respiration, and what's important is this maximal respiratory capacity and reserve capacity. Reserve capacity is important because it tells us about how much reserve the mitochondria has. And when this goes to 0, that means that at baseline, the mitochondria is working at its maximal capacity. We know the mitochondria is very sick because it has no reserve, and the cell is probably going towards apoptosis. So we did our first experiment, and this is another example of how sometimes you get unexpected results, but you maybe do something with them and you figure out what's going on. Of course, there was the idea that kids with autism had mitochondrial disease where the mitochondria should work very, very poorly. So we thought it would be a simple experiment. We put these cells in the seahorse and we look at their respiration rate, which is on this axis, and they'd be below the controls, right? Because mitochondrial disorders are thought to be when the mitochondria is working really poorly. And lo and behold, we found the opposite. They actually had high respiratory rates. And what we found is that when we added oxidative stress, they were actually more sensitive to oxidative stress, that they lost their reserve capacity much faster than the controls. One of the things we said, everything in autism is really subgroups, and maybe is there a subgroup driving this? And the answer is yes, and we found this subgroup of mitochondria, about a third, that were driving this effect. And when you plot it out, it's really interesting. We can look at reserve capacity, just to speed along. We can see that the cells from autism, which normal mitochondrial function, overlap the controls almost completely, whereas the ones from the cell lines that had this elevated mitochondrial function was actually twice. All the respiratory rates were twice that of controls, for really distinct pattern. We also replicated this in PBMCs from patients to make sure it wasn't just an artifact of the cell lines, and we actually found that about 25% of our patients had this characteristic of very high respiratory capacity. And then we compared them to kids with mitochondrial disease because we thought, well, maybe this is different than classic mitochondrial disease. And you can see those are the blue here, and compared to the red, that for the most part they don't overlap. So this seems to be a unique type of mitochondrial disease. Then we were able, in a recent study that we did with Dr. Zimmerman, to link this to neurodevelopmental regression in kids with autism. So this is a study, we looked at neurodevelopmental regression. This is a seahorse plot, and we can see that the maximal respiratory capacity is much higher in those that had neurodevelopmental regression. And further work, we've shown that actually environmental factors, prenatal air pollution, is proportional to this elevation in respiration in those that have developmental regression. So it actually seems that environmental exposures prenatally may actually have an influence on programming the mitochondria for this effect. We've showed in siblings that this is unique to the one with autism as compared to the typically developing siblings, that they have this high reserve capacity here. And so this comes our new data. So we wanted to replicate the data to say, okay, is this really something we see in kiddos with neurodevelopmental regression? So we got a new cohort, and we looked at kids with neurodevelopmental regression and no neurodevelopmental regression. And there actually is a sex effect too. So that's why there's so many lines here. It actually ends up that it seems that females have higher respiratory rates than males. But what you can see here, this is the kiddos, the females with neurodevelopmental regression, and these are the females without neurodevelopmental regression. So you're still seeing that distinct pattern, and these are the males with neurodevelopmental regression without neurodevelopmental regression. And so these are all kids with autism. So then we compare them to the typically developing siblings. So these are siblings, and we see those without neurodevelopmental regression. They have essentially very similar patterns, respiratory patterns, as compared to their typically developing siblings, whereas those with neurodevelopmental regression have this distinct pattern as compared to their typically developing siblings. Again, suggesting that this is a characteristic of neurodevelopmental regression. But the thing that we didn't expect is when we looked at, let's see, I think it's the next slide. When we looked at parents. So one of the things that we did, we just decided, well, let's take a look at parents and see if this is something that runs in the family or not. And it's kind of an interesting story. What we first did is we said, well, is this a phenomenon? The first thing we did before we did any analysis or anything is we just took the graphs, the plots, and we sorted them into those that had high respiratory rates and low respiratory rates. And it was almost 100%. That we found those with neurodevelopmental regression and those without. And we said, well, what would we use? Could we use some type of control? Maybe we use the parents, because the parents shouldn't have this abnormality. We'll say, which are the ones that have high respiratory rates relative to the parents? And then we sorted them that way, and there was no results. We said, what's going on here? So then when we analyzed the data, what we found in this is these are not the kids, these are the parents of the kids. So the parents of the kids, and again, female, male, differences, these are the parents with kiddos with autism with neurodevelopmental regression, and these are the parents with kids with autism without neurodevelopmental regression. And recapitulating the same phenomenon that we saw in the kids with neurodevelopmental regression. So most of these kids, one of the mysteries of mitochondrial disorders is that many times we do not see any clear genetic abnormality that's driving it. Sometimes we do, but for the most time, we do not. So the question is, how is this inherited from parents to children? And so that's kind of our next step to try and figure out, but this may be something that may be in the environment that may be affecting the parents and the child. So with that, I'll thank you for your attention. Again, I want to remind you, as Pramila showed, we have these special issues that we published papers in last year, and they're still open for further submission of papers, and it would be really great to get more papers in here so we can have a nice completed special issue from our Synchrony work. We have a special issue, and 2 are in the journal Personalized Medicine. This one is a personalized medicine approach to the diagnosis and management of autism spectrum disorder, but beyond genetic syndromes, edited by myself, Dr. Bowles, Dr. Rose, and Dr. Rossignol. And then another one on epilepsy that's edited by Dr. Gatanis and myself and Jeffrey Lewine. And then we have another one in the Journal of Translational Genetics and Genomics on the genetic basis and treatment of psychiatric disorders. So if you have any good papers, please consider these as a place to submit them to. Thank you.